Today we will discuss about therapeutic approach to reduce HIV infectivity using CRISPR technology. So in this case or in this paper, researchers has used T cells where they have modified the MOCs, and the MOCs are generally found in the endoplasmic reticulum of the cells, and it helps in the post-translation modification, or more specifically it helps in the N-glycosylation. And in order to produce more HIV virus, it needs to acquire its native structure. And in order to acquire the native structure, they need to go through this calnexin and calreticulum cycles, which that helps in acquiring the native structures, which occurs in the N-glycosylation endoplasmic reticulum. That topic I have already covered many times in our previous videos. So here, what the researchers have used, they modified the MOX knockout in the T cells, so that when the HIV virus goes through when the HIV virus infected the T cells, they will not acquire their native structures, and therefore the HIV virus that are reproduced by those modified T cells can produce ineffective HIV virus. So that will lead to the reduced infectivity of the virus. And another important thing that they have taken care of in this paper is that the CRISPR MOCs will be activated in those cells that are already affected with the HIV. So this will reduce the non-specific editing of this MOX virus on the healthy cells. So let us understand how this approach works. So first is this HIV that has the infectivity. So it can interact with the CD4 and CCR5 receptors, and uh, that helps in the fusion of this HIV virus. So once the HIV virus is fused, then through the reverse transcription process, the viral DNA is integrated into the host genome, and thereafter the CRISPR HIV will, since it is modified in the form of CRISPR HIV, the CRISPR and HIV will form a DNA excision bond, and this will results in the cleavage of any of this CRISPR HIV, any of the further CRISPR HIV, any of the further HIV infections of these cells. And simultaneously, what will happen? The integration that has already been occurred, since these kind of cells are also CRISPR MOX knockout. So when the transcription and that is the conversion of this DNA to this RNA, viral RNA, uh, and thereafter the translation occurs. So during the assembly process, it has to go through the endoplasmic reticulum, and since this cells does not have the proper MOX genes, the proper glycosylation of GP120 and GP41 will not occurs, and the improper abnormal glycosylations occurs in the MOX negative cells. And these virus core structures, abnormal virus core structures, will move out of the cells, and that has the reduced infectivity or without any infectivity. So this is the approach that they have used in this paper to make their point that this CRISPR technology can be integrated with this HIV and MOX on the T cells of the patients or the immune cells of the patients, so to reduce the HIV infectivity on those patients. So how they will make sure that the CRISPR MOX or the CRISPR MOX will occurs only in those cells that are affected with this CRISPR HIV. So they have integrated the cells, or they have designed this CRISPR MOX in a way that it will the CRISPR MOX will start to transcribe or translate only when these CRISPR HIVs are formed. Or when the CRISPR HIVs are formed, then it will deactivate, or this CRISPR HIV will deactivate the MOX gene, or we can say edit the MOX gene, so that the MOX gene will not be, so that cells will be divide of the native MOX. So I hope you got some basic or brief overview about how this therapeutic approach can be used to reduce HIV infectivity. If you need to understand in detail about this paper, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.